We have the pleasure of hearing from Nayan, who's a postdoc at NCBS, and he'll tell us about some of the work he's been doing, regarding very similar things, but from an experimental perspective, right? So please take it away, Nayan.
Thank you, Praful. Okay. Thank you, everyone. Yeah, so today I'm going to talk about a very simple compartmental system, which is capable of what you call proto-metabolism. So just to dive straight into it, this is just a basic schematic of the origins of life problem on Earth. On one hand, we have habitable world. On another hand, we have the diversity of life, all the way starting from Luca, if you may. And in between, there are all these steps of complexity. So like one can start with prebiotic synthesis in a habitable world, and then starting from prebiotic synthesis of simple molecules, and one would expect to get at some point polymers or other supramolecular structures like the one Doron was talking about, micelles, vesicles, and others. And then at some point, all of this would be integrated to form something called a protocell, which has the basic functions of a cell, and then through evolution, one would get something like a modern cell. This is usually studied in two different approaches, like in a top-down and a bottom-up approach. And regarding our take currently on this, we have sort of identified three broad themes of problems in this entire origins of life schematics. So I'll just briefly describe them and then delve into the system. So the first one, what I call, is a compatibility problem, which literally is so in prebiotic chemistry synthesis, there are ways to synthesize the different precursors of biomolecules like sugars, amino acids, nucleotides, lipids. But most of the time, these chemistries are not compatible with each other. So that's what we call is a compatibility problem. The second one, what we call is a discontinuity problem, is that starting from simple molecules, as more complex molecules are synthesized, these sets of reactions are usually not coupled to structure formation or boundary formation. So to us, discontinuity problem is one in which one can start with simple reagents and can simultaneously get complex chemical reactions and structure formation in one part. So that's the discontinuity problem. And the third one, what we call is the bioinorganic problem, which has also been discussed, is basically modern life incorporates different inorganic elements in its catalytic sites, which are quite crucial throughout the tree of life. But most of the simple forms of protocells and other are not compatible with different concentrations of inorganic elements. So at some point, our models of origins of life need to incorporate the inorganic elements throughout its history. So this is what I call the bioinorganic problem. And now to describe just a bit about a model, a simple model for a protocell would be something like this, which is the chemoton model proposed by Ganti. So one starts with some food set, which is not alive, it's just food set. Starting from this food set, one would get an emergent metabolism cycle, and this metabolism cycle would form the uh, structure in which it would contain the metabolism cycle. So that is the structural cycle, and also an information containing cycle. And all these three cycles would be coupled to each other. And of course, through this entire process, waste would also be generated into the environment. So this is a simple model, a simple structural and chemical reaction network model for protocell. Now, what is the type of food that one would like to start with? So one can argue at a different lens regarding the molecules one would start with as a food, but at least at an atomic level, we know more or less what uh, most of life is made of. So the elements that are present in green is are the ones necessary for all life as we know it. The common acronym is called SNOPS. So basically, carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur are the most essential elements. Like they take up to like 98% of almost all living systems that you know. And then they in different hierarchies, we have transition metals, which are beneficial for all organisms like iron, cobalt, nickel, copper, zinc. And then we have other elements like sodium, potassium, magnesium, which are also beneficial for all organisms. And then some trace elements. So this is kind of the atomic soup one can expect to start with, and certain molecules which are made of this atomic soup. So to dive into our system, we started with a simple four molecule system which has SNOPs incorporated in it and metal catalysts. For metal catalysts, we chose iron and molybdenum, which is known in extant biology as a good for redox reactions and even nitrogen fixation throughout serving different catalytic roles. So we start from a simple mixture of these molecules in which formaldehyde is the sole carbon source. So this is the formaldehyde, which is the sole carbon source. And then starting from there, we optimize the conditions at some point at some concentration of these starting materials. We get these microsphere particles which emerge in our system, which can be observed through microscopy, and they are bright blue in color, and due to the presence of reduced molybdenum in them. And initially optimize the system in order to find the roles of the different ingredients that we have used in order to form these blue microspheres. And then we went ahead in order to characterize the different properties of them. And to start with, we characterized its atomic compositional information, so to speak. And the technique we used is EDAX, which basically shoots X-rays at a sample in order to know the atomic composition of that sample. And here in this plot, you can see in green is the atomic composition of the initial solution, the well-mixed featureless solution. And in blue, the composition information of the microspheres is plotted. It shows that the stoichiometry is distinct from the solution from which it phase separates out. And and also when we plot the different enrichment factor of the different elements in our microspheres, we do see signs of selective atomic sequestration in our microspherical simple sample, suggesting it's not just pure precipitation process which is giving rise to these blue spheres. In order to then he characterized its physical properties. So from microscopy, they're liquid-like with soft and adhesive interfaces with signals of surface tension. In order to quantify some of this softness and adhesiveness, we subjected them to force indentation measurements. Here, the control is a glass surface, and on right side, this is our sample, which is subjected to force indentation. And from these measurements, we do indeed see these microspheres are quite soft and they are quite sticky also, and we can compute an addition energy from this force measurements for them. So indeed, they're soft and sticky, they're liquid-like, and we wanted to image their interiors also. For that, we ended up doing a transmission electron microscopy and also different chemical treatments in order to partially visualize their interiors. And yes, it seems like, yeah, the microspheres, the interiors is quite heterogeneous and they're compartmental in nature. They can take up molecules and can to various degrees. So they seem to be semi-permeable in nature also, as you can see from this image. Further, high resolution bursting experiments in aqueous environment of these microspheres revealed their liquid-like nature, in which we can expulse their heterogeneous lumen from these microspheres locally by perturbing them with lasers in order to burst them. As you can see, they ooze out the liquid material from inside them. So yes, they do have a boundary with a specific heterogeneous interior, which is separate from the bulk solution outside. And even further high resolution TEM cross sections and SEM images also revealed to have some kind of a boundary like shell material along with heterogeneous interior with their different electron densities, suggesting different organic and inorganic species incorporated inside the interior of these microspheres. And very high resolution TM images at nanometer scale of the surface, the shell of the microsphere, do reveal a mixture of a mixture of hybrid architecture in which one can see there are small crystalline, nanocrystalline domains on the shell of the microsphere, which form these ordered moire-like patterns. And also there are amorphous regions which resemble brush-like long amorphous regions on the shell. So overall, the shell seems to be a hybrid organic-inorganic mesh-like material. So this is about the structural and the compositional characterization of these microspheres. And then we wanted to characterize some of its dynamics and other functional features. And for that, we started by characterizing its optical density. And left side here, it's a control. Control basically has all the elements that I mentioned in the beginning, all the ingredients except the carbon source, which is formaldehyde. And this will be our control more or less for rest of the talk. And in that, in the OD measurement over time across wavelength, we do see there are some signatures of small nanoparticles in the system, which is generated from the reduced molybdenum iron complexes, but there's nothing else, so to speak, in this sample. But contrasted that with our microsphere solution as a function of time, we do see there are different bands of particles of different size that start to show up in your OD measurement. So this is a population level measurement. And if we fix the wavelength, so there are few peaks. We did characterize most of the peaks in this solution. And there are few peaks. If we fix, let's say, one of the more important peaks, which is, so there are two peaks which are more important. One is at 350 or the other is 840. And if we plot the OD as a function of time as one of these fixed peaks, like how one does for bacterial solutions, we do see a supralinear increase in the OD in the initial time scale um, in our microsphere solutions. And this supralinear increase in OD is also matches with the supralinear sigmoidal like increase in the mass, total mass of the microspheres generated as a function of time. So both of these together does point towards some autocatalytic signatures in the formation of these microspheres starting from the simple salt solution. In order to test that, we did one simple serial transfer experiment in which we let the microspheres grow for some time and then transferred a small fraction of it to fresh substrates and again let them grow for some time and again transfer them to fresh substrate and repeat it around 4 or 5 times. And then for all of this, measure the OD. And we do see that as a function of serial transfer, the lag time and also the lag time decreases and also the optical density increases too, which shows us initial signatures of autocatalytic like behavior in our system of microsphere formation. And now, but this OD and mass measurements are measurements at population level. We wanted to check what is happening at a microscopic level to the microspheres as a function of time. So the dynamics of them as a function of time. And then so we did single particle tracking of these microspheres over time. And we do indeed see that, yeah, not only like new microspheres form over time, but the microspheres also grow in size over time at a single compartmental level. And when you plot the statistics of these microspheres, we do see that, yeah, they kind of tend towards becoming more and more polydispersed. And also clearly there is a growth rate dependence on the size of the microspheres, which suggested us that it's not a simple precipitation process through which it's growing, but more or less a surface catalyzed mechanism. In order to test it slightly better, we fitted this the initial time scale growth. And typically, just to give an example on radius dependence on various types of mechanisms of growth, usually for bulk condensation, the R cube is proportional to time. And for division limited process like crystal growth, it's R square proportional to time, the scaling. And for surface reaction or surface catalyzed ones, it's R, which is proportional to time. And we do see that in our microsphere scaling fit, we find two regimes: an initial very early regime in which it's R cube proportional to time, and it's mostly gives a sign of volume controlled growth, which makes sense because it's the initial nucleation is governed by condensation of these molybdenum iron nanoparticles. So it's a bulk condensation process. So it's volume limited. And also, and then the growth process seems to be the R almost proportional to T, and which points towards a more surface catalyzed mechanism for the growth of this micron-sized sphere. So starting from nucleation and then growth seems to have slightly different mechanisms, which is kind of compatible with the chemistry that we are working with. So that is about the function of growth. And then we wanted to also characterize the atomic composition and information in a dynamical sense, what is happening over time. And so when you analyze it, we do see that the atomic composition of these microspheres increases over time in a supralinear manner and then almost uh, reaches saturation beyond around 4 hours. And when you plot the fold change as a fold change of each element in terms of its composition as a function of time, we do see most of them except carbon reaches a steady state after around 4 hours in the microspheres. So it reaches a steady state except carbon, and the growth beyond this time scale seems to be only governed by accumulating more and more amounts of carbon, which pointed us towards that, okay, they may not be just structural microspheres which are just accumulating carbon, but they may also have some functional features. And so we wanted to see, characterize the chemistry which is happening in the entire system, both in the bulk and also in the microspheres. And in order to characterize the chemistry, first we did simple experiments in which we tracked the pH and also the heat released in calorimeter of these entire samples. And we did see there's a sustained proton release and also heat release over across 3 weeks in our microspherical samples as compared to the controls, which lacked the formaldehyde in the solution. And so this gave us boost to check out to carry out high throughput analysis of the molecular information in this system. And so we uh, uh, investigated the system using LCMS and NMR. And so I'll just go through the results here. So LCMS, time-dependent LCMS measurements did show us the synthesis of numerous different molecules over time, starting from 50 initial 2 days, over 20 days, and up to even 42 days. And then and also combining with NMR, we do see that this substrate peak, which is the peak of formaldehyde gets consumed in our system over time, and we also get certain different products which appear in the system over time. And we have characterized many of these products. So mass spectrometry also shows a similar result of complexification of the chemistry starting from formaldehyde over time and to form a set of different molecules. And indeed, so the substrate is formaldehyde in our system, which is getting consumed. And this was done with labeled formaldehyde, so 13C formaldehyde, and slightly better analysis of of certain regions in the NMR spectrum, which points towards signals corresponding to longer chains. We do see in both the 13C NMR and also proton NMR, we do see progressive synthesis of alkyl chains or longer chains in our system as a function of time, and their abundance also increases up to an order of magnitude over time. So yeah, so not only like small molecules are being synthesized in our system, but also there are certain signatures of longer chains that are being synthesized in a progressive manner over time in this chemical system. And uh, and when you analyzed the protocells, which are the microspheres separated from the bulk solution, which is the supernatant, we do see that there are more molecules present in the microspheres as compared to the supernatant solution around it. And also there are longer molecules which are more present in the protocell. So molecules of larger mass, if I may. So molecules of larger mass are distributed more in the compartmental space as compared to the bulk solution around it, suggesting us that, yeah, indeed, these compartments do act as crucibles of organic synthesis and synthesizing more and more larger molecules and longer chains over time. Slightly more careful analysis of this organic space of products from a biomolecular uh, lens gave us that, indeed there are various biomolecule-like species which are being synthesized in our system over time, like the there are amino acid-like and sugar-like and amplified-like molecules which are synthesized, and a simple classification like this Van Crevelen classification on the uh, mass spectrometry data gives us a population level idea of what are the different types of molecules that are being synthesized in this messy chemistry system. And so overall, we um, are constructing a reaction network which kinds of points towards how, starting from formaldehyde, which is the sole carbon source, one can get the different types of molecules and the biomolecules that one sees in the data. And broadly speaking, the entire system is driven by a redox engine between iron and molybdenum, and iron and molybdenum combined together simultaneously oxidizes and reduces formaldehyde in different proportions. And starting from there, so these are the reduced branches of formaldehyde, and so these are methane diols, methanols, and other polyols, uh, and this is the oxidized branch of formaldehyde, which is formyl or formic acid. And starting from there, one can uh, get different synthesis of different biomolecular classes. I can go through it in detail also in the discussion section, how one can get different biomolecular classes. But this initial reduction and oxidation, which happens in a very fast time scale, we also have an idea about the time scale of all of these processes. Yes, so the initial redox process is important for the formation of the compartment. So even the compartment does not form if there is no formaldehyde in the system. So starting from formaldehyde, one generates a pool of oligomers, which then interacts with the metal nanostructures and then phase separates out to form this hybrid shell. So that's the way the compartment also forms, and then downstream chemical synthesis also happens overall. So and to move ahead, we then explode our system in order to observe or have experimentally see certain signs of propagation, if there are some signs of propagation in our system. And so when we were doing this high resolution imaging and timeless microscopy, we did many times see that there are signatures of propagules which are present inside these microspheres. You can also see them here. And sometimes during our busting experiments, many times these propagules would come out. And even in our long-term EM experiments, we could see in high resolution that there seems to be some budding like smaller microspheres inside the bigger microspheres. So we thought maybe we can facilitate, we can use that process in order to experiment on some rudimentary propagation. So what we ended up doing is we grew these microspheres for some time and then blasted them completely. So these are the 3, 4 microspheres, and you can see all of them are destroyed, and they have churned out some nanoscopic propagules here roughly all around. And then we put fresh media into it, and then we uh, wanted to see what happens to this propagules over time. And we could indeed see that yeah, these propagules do become bigger, grow over time in fresh media. And we wanted to uh, quantify it also in terms of its elemental composition. And when we did that experiment, we saw that, okay, there's some variations, but it's similar. The composition and information of the elemental composition information of these propagules, newly generated propagules, which we're calling P1, is similar to the atomic composition of our original microspheres, which we're calling P naught. And this is being investigated more now. And so then overall, we have this picture. So that's the summary. We have a simple aqueous mixture of 4 salts, starting from which we have a mechanism through which so formaldehyde gets oxidized and reduced, and a few more steps are also incorporated, and then it generates an oligomeric pool, and simultaneously the molybdenum and the iron also generates this POM clusters, which are well known in the field. And overall, these POM inorganic clusters and the polyols that are generated from the formaldehyde, they interact and they phase separate out to form these microspherical structures, which then feed back to the in a catalytic manner to its growth and expansion. So this is our current model for our system. And just to end on a note of on natural occurrence of similar chemistries. So all this is being done in the lab. And but we found that 1 year back at least, so there was this study in which they found blue sponges in the ocean, and these sponges were blue because of certain small blue molybdenum-rich particles being present in them. And in so on left hand side we have image from that paper in which they show different TEM images and also the microscopy images of the blue microspheres present inside the sponges. On right hand side, we have the microspheres found in our lab. And not only that, they also report certain yellow redox states associated with molybdenum also found in the sponges, which also we find in the lab while we are optimizing for our blue microspherical solutions. So the structures that we see in the lab and the structures they find in the ocean sponges seems to be strikingly similar. But not only that, they have the data for the atomic composition of these microspheres in the sponges. So within sponges is the atomic composition of that. And when you plot that data against our lab-grown microspheres in a time-dependent manner, we do see that they do share striking similarities to the structures found in the oceans. And so overall, so this is where I'd like to end my talk. And yeah, so we start from a simple high entropy mixture which does not have any feature, that doesn't have any feature. And then over time, we form these growing spherical compartments, which also does metabolism. And you can read the our preprint. It's out on bioarchive. And and so just to come back to the initial point we place, so in this context, we place our macrosphere somewhere here in the prebiotic synthesis uh, step in terms of concentrating. So some prebiotic chemistry happening and concentrating certain molecules. But not only that, we also propose it as a bridge towards the three different problems, which is the compatibility problem. And now, so because in our chemical mixture, we do have indication of synthesis of different, so to speak, incompatible classes of biomolecular species like lipid, like amphiphyte-like or amino acid-like or sugar-like in one system. And it also is a step towards the discontinuity problem, because starting from just a featureless solution, we get simultaneous synthesis of a chemical reaction network and a compartment and a feedback between the two, which then complexifies both the compartment and the chemical reaction network. So simultaneous synthesis of the reaction network and also the boundary. So which is the discontinuity problem. And also it may provide a natural route towards concentrating inorganic molecules in the beginning and then slowly over time through dilution and other and maybe other ways they will the inorganic materials will be incorporated as catalytic functions in modern systems like protein-based systems or RNA or DNA-based systems. But even though the function of the material will still be similar, that is, it will play a catalytic role in the reaction network that it will be inhibiting, so to speak. So so I'll end my talk here. So thanks to my PhD advisor Shashi and NCBS, where I did it, and I'm working now as a postdoc on this problem, and the autocatalytic reaction network seminar organizers Alex, Nicola, Praful, and Wei Xiang. Thank you.